Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, GenScript USA Incorporated, I'd like to welcome you to MonoRab, generating rabbit mAbs with better affinity and specificity for challenging targets. My name is Elizabeth Lamb, and I'll be the host and the moderator for today's event. At this time, I'd like to introduce our presenter for today. He is Dr. Liusong Yin, Associate Director and Head of the Antibody Department at GenScript USA. Dr. Yin received his PhD from the University of Massachusetts Medical School, specializing in antigen presentation and CD4+ T-cell epitope selection. He then joined Pfizer, specializing in the immunogenicity of therapeutic biologics. Dr. Yin has extensive experience in antibody drug discovery, from lead generation to lead optimization, especially in the field of DNA immunization, high-throughput FACS screening, ELISA kit development, and anti-idiotype antibody development for antibody drug PD/PK studies, as well as antibody humanization and affinity maturation. In this webinar, he'll give an overview of rabbit monoclonal antibodies, then talk about MonoRab from GenScript to generate those rabbit mAbs, after which he'll give several case studies on those. With that, I would like to welcome you, Dr. Yin. The presenter ball is yours.
Thank you, Elizabeth, for the introduction, and thanks everyone for attending this webinar. As mentioned, I will talk about MonoRab technology generating rabbit mAbs with better affinity and specificity for challenging targets. This is the outline of my presentation. I will first give an overview on rabbit monoclonal antibodies, and then talk about GenScript's MonoRab platform to generate rabbit mAbs. I will share several case studies for applications during the presentation and give you some facts about GenScript, and I'm happy to answer your questions at the end of my talk. So when it comes to antibodies, researchers always have concerns for how well their antibody work in their experiments. Sometimes what troubles researchers are the difficulty to obtain an antibody to begin with. Antigens such as small molecules cannot trigger immune response in mice and rats, and for some cases, antibodies are required to distinguish minor differences from antigens, such as post-translational modifications or protein sequence variations as small as one amino acid. Usually, the successful rate of generating marine antibodies is on the lower end. Application-wise, certain properties such as sensitivity and specificity are desired in particular for detection purpose. For the above-mentioned scenarios, rabbit monoclonal antibodies can be an alternative and sometimes even better option. So rabbits are well known to mount a strong immune response against foreign antigens. Rabbit antibodies are extremely useful when the antigens are not immunogenic in mice. The superior features such as high affinity, high specificity, high diversity allow rabbit monoclonal antibodies to yield excellent results in applications such as anti-idiotype and for in vitro diagnostic antibodies. The affinity of rabbit monoclonal antibody can reach picomolar level, while marine antibodies are typically around the nanomolar level. Rabbit mAbs exhibit low cross-reactivity, and they are able to distinguish very similar proteins and sequences. Hundreds of rabbit mAbs are used as reagent by researchers to study biological functions of various proteins, as well as biomarkers to evaluate clinical candidates and their performance in clinical trials. So the natural rabbit antibodies in IgG format is shown on the top of this slide. Rabbit antibody is somewhat simplified compared with mouse and human antibody. No rabbit IgG has been found so far. The abundant class in rabbit serum is IgG, with a serum concentration about 5 to 20 mg per ml. Unlike IgG from other species, there are no subclass in the rabbit IgG, only one IgG isotype for rabbits. Rabbits of the commonly used New Zealand wild strain have about 90% IgG carb K1 and about 10% IgG carb K2 and less than 1% IgG NAMTA antibodies. Rabbit IgG is also strongly different from that of mouse and human. Rabbit IgG tends to have fewer amino acids in the N-terminus and in the DE loop. It also has extra disulfide bonds in the variable region. Rabbit has a different mechanism for generation of its antibody repertoire, as shown in the molecular mechanism of sequence diversification in rabbit antibody repertoires. All this contributes to the high diversity, high affinity, and high specificity of rabbit mAbs. Currently, there are three major platforms or technologies for generating rabbit monoclonal antibodies: phage display, hybridoma approach, and single B-cell based methods. In phage display platform, rabbit antibody heavy chains and light chains were amplified and randomly paired to form new rabbit sequence to screen for positive binders. In hybridoma approach, rabbit B cells or splenocytes are fused with melanoma fusion partners, and subnatant of resulting hybridomas will be performed for various positive and negative screening to identify specific positive clones. In single B-cell based approach, the antigen-specific B cells are sorted into microvials as single cell and the antibody sequence is extracted. Recombinant antibody will be purified for multiple characterizations. To summarize the key aspects of generating rabbit mAbs using hybridoma, single B-cell, and phage display, three different approaches. In terms of turnaround time, nature of chain pairing, antibody affinity, diversity, and project cost. So single B-cell cloning technology can have lowest turnaround time if the platform is very well established. Phage display relies on the proper transcription, translation, folding, assembly, and random pairing of light and heavy chains, and therefore the chain pairing is not native, will result in relatively low affinity and diversity of obtained rabbit mAbs compared with hybridoma and single B-cell approach. In cost-wise, single B-cell platform requires most investment and highest cost. With this quick comparison across different platforms, combining quality, cost, and turnaround time, the hybridoma technology is still the most cost-efficient approach to generate high-quality rabbit mAbs. So next, I will spend some time on introducing GenScript MonoRab platform for rapid mAb generation with the hybridoma approach. So as mentioned before, the hybridoma approach is the most cost-efficient way for generating rapid mAbs. GenScript has extensive experience with hybridoma platform over the past 12 years. For MonoRab, mouse melanoma cells are used to generate rabbit hybridoma, and the antibody is fully native rabbit monoclonal antibody. Highlights of our platform included the improved fusion efficiency and hybridoma stability, as well as combining antibody sequencing together with recombinant antibody production. As a result, MonoRab platform delivers rapid mAb sequence to avoid unstable clones, which allows better antibody reproducibility. We have used MonoRab technology to generate rapid mAbs against a wide range of targets, such as antibody drugs and endotype antibodies, small molecules, receptors, epigenetic modified antigens, and post-translational modifications. So this shows you the milestone-based workflow for MonoRab. It includes immunogen preparation, followed by animal immunization, cell fusion and screening, subcloning and sequencing on the subclones. The last step is recombinant rabbit mAb production. From immunogen preparation to purified antibodies, it can be as short as 4 months. Highlights including antibody validation such as epitope binding is provided for free, and the antibodies we generated for clients belong to you. You own the IP, no licensing or royalty fees will be offered. So a more detailed service specs are listed on this slide. There are 4 major milestones and 3 decision gates in the process. The project begins with material preparation, including protein antigen expression and peptide antigen synthesis and conjugation to carrier proteins, as well as necessary counter-screening material preparations. The immunization will be performed with optimized adjuvant and a schedule to achieve optimal titers. Rabbits with top titers will proceed to cell fusion and various screening steps. After positive parental clones are obtained, 1 round subcloning will be performed and the positive supernatant will be delivered for your in-house test. Antigen V-region sequencing will be performed for desired clones and the sequence will be delivered to avoid clone stability issues. Optional recombinant rabbit mAb production can be performed to deliver purified rabbit mAbs for various applications. So here I summarize the MonoRab technology advantage. Optimized fusion partner and improved fusion efficiency, which allow high fusion efficiency of 1/1000 to 2000 B cells. Then super screening platform is also integrated. The instable hybridoma is addressed by early stage antibody sequencing together with recombinant antibody production. Rabbit mAbs can be superior to mouse mAbs in many aspects as discussed before. For some particular applications, such as ELISA, Western, IP, FACS, and so on, rabbit antibodies can be used as an alternative approach where there is no luck generating marine antibody with high affinity and better specificity. And for some other applications for detection, such as anti-erythrotype for PK studies and IgG detection antibodies for IVD studies, rabbit mAbs can improve the success rate compared with mouse mAbs. So in the next few slides, I am going to show some case studies and applications of rabbit mAbs. GenScript has produced rabbit mAbs which are highly specific, sensitive, and immunoreactive against a wide range of difficult immunogens such as small molecules, nanobodies, which are single-domain antibodies, membrane receptors, and antibody drugs. Applications including pharmacokinetic assays and immunogenicity, which are anti-drug antibody assays, as well as ELISA kit development and in vitro diagnostic detection can all benefit from this MonoRab platform due to the various advantages of rabbit monoclonal antibodies. So to summarize, high diversity, high affinity and specificity, and a novel epitope recognition. So in the first case study, we generated rabbit mAbs against a small his tag and compared the sensitivity with mouse anti-his tag antibodies. The LAMC is the best mouse anti-his tag antibody coming from our numerous screening, and the LAMC and B are both rabbit anti-his tag antibodies. As you can see from that, the 2 rabbit anti-his tag antibodies showed better sensitivity compared with the mouse counterparts. In the second case study, we generated several rabbit mAbs against the flag tag in C-terminal, N-terminal, and the middle of fusion proteins. And again, compared with mouse counterparts, we have demonstrated that these rabbit anti-flag tag antibodies have higher affinity in the range of 10 to the minus 11 molar and have lower reactivity with other tags. As you can also see from the figures that all 3 rabbit anti-flag tag antibodies showed much better sensitivity compared with mouse anti-flag tag antibodies, which are great for Western blot and immunoprecipitation assays. In this case study, we developed a panel of rabbit mAbs against single-domain antibodies, which can be used as secondary antibodies for screening nanobodies against a variety of targets, as well as for PK studies of nanobodies. As we all know that generating specific antibodies against the nanobody is really challenging due to the low antigenicity of nanobodies and the similarity with antibodies from other species. In this campaign, more than 20 rabbit monoclonal and anti-nanobody antibodies were generated by immunizing with nanobody heavy chain. The affinity are very high in the range of 10 picomolar range. For the best two anti-nanobody rabbit mAbs, they universally recognize 20 random camel single-domain antibodies, but show no cross-reactivity with mouse IgG, rat IgG, rabbit IgG, goat IgG, chicken IgY, and human IgG. So which means they are very specific. For this highly specific anti-nanobody rabbit mAbs, we performed re-region sequencing for 8 of them and expressed them in mammalian cells. All of the 8 rabbit mAbs were successfully cloned and expressed, and the binding activity of recombinant rabbit mAbs against nanobody have been confirmed by ELISA. So small molecules are a special group of antigens which are extremely difficult to generate specific antibodies. The challenge comes from the fact that small molecules are known to be very low antigenic to elicit robust immune response for antibody generation. And also, the structure of small molecules is usually not complicated enough to identify specific antibodies. In this case study, we took the advantage of MonoRab Platform and have achieved great success in generating rapid anti-ractopamine antibodies. The antibodies generated have very high affinity in picomolar range. As you can see from the figures with both indirect ELISA and competitive ELISA, the representative anti-ractopamine rapid monoclonal antibodies showed EC50 and IC50 about 1 nanogram per ml and 0.05 nanogram per ml respectively, means they are very specific and they have very high affinity. So we further evaluated the specificity of anti-ractopamine rapid mAb against several small molecules with similar molecular structures with ractopamine. As you can see from the comparative ELISA results, the cross-reactivity rate of rapid anti-ractopamine mAb with suboptimal is only 0.03%, and with clenbuterol is also about 0.03%. This data showed that our MonoRab platform can generate rapid mAbs with high specificity with low cross-reactivity with similar compounds. So in the fifth case study, we evaluated the feasibility of generating rapid mAbs against receptor proteins. Glucocorticoid receptor was used as the immunogens to immunize rabbits, and we have successfully obtained a panel of rapid anti-glucocorticoid mAbs. We performed re-region sequencing for 3 of them and expressed in mammalian cells. All the 3 clones were successfully cloned and the recombinant antibodies were purified. The sensitivity and affinity of the 3 recombinant expressed rapid anti-glucocorticoid antibodies were validated by ELISA, as you can see from the bottom right figure. Anti-ETO-type antibodies are must-have tools for antibody drug discovery. There are 2 major applications for anti-ETO-type antibodies. It can be used to directly measure the antibody drug level in patient samples in pharmacokinetic assays, shown as PK assays. It can also be used as a positive control or reference standard for anti-drug antibody in immunogenicity assays. Rapid monoclonal anti-ETO-type antibodies are recommended to be used as reference or positive control in immunogenicity assays. As we discussed, rapid monoclonal anti-ETO-type antibodies are perfect for PK studies due to its high affinity and high specificity. So there are 3 different types of anti-ETO-type antibodies. The first one is antigen blocking or paratope specific, which inhibits the antigen binding when bound to the antibody drug and can only detect free antibody drug. The second one is non-blocking or non-paratope specific, which does not inhibit antigen binding and can detect total antibody drug including free, partially bound, and fully bound. The third one is not that common, which is complex specific, can only detect bound drug. An important aspect of anti-idiotype antibodies is the ability to perform guided selection during the development process to isolate anti-idiotype antibodies with application-based specificities. Since 2009, GenScript has been specialized in making anti-idiotype antibodies to support our customers' PK, PD, and immunogenicity evaluations of antibody drugs. Our record of success rate is 100% with more than 200 projects. We have developed anti-idiotype antibodies with high specificity that only recognize antibody drug but not ISAP control and total IgG in human serum. I will give you a case study on using our MonoRab platform to generate anti-idiotype antibody for PK studies. In this case study, the goal is to generate anti-idiotype antibodies against anti-PD-1 antibody drug, pembrolizumab, or the brand name is Keytruda. For PK studies of Keytruda, rabbits were immunized with Keytruda and a panel of rabbit anti-Keytruda idiotype antibodies were obtained, and all of which have high affinity and high specificity. For the representative clone 90G12F8, it shows an ELISA EC50 of 5 nanogram per ml in indirect ELISA, IC50 of less than 20 nanogram per ml when blocking the binding of human PD-1 to Keytruda. We next compared the binding affinity and ligand blocking activity of rabbit clone 90G12F8 and several mouse anti-Keytruda clones. As you can see from the data, even though the binding activity of rabbit anti-Keytruda antibody is comparable with mouse counterparts from the EC50, the blocking function of rabbit mAb is much better than mouse monoclonal antibody. We also checked whether rabbit monoclonal antibodies produced using stabilized hybridoma cells, and we checked the binding affinity. Besides affinity ranking by Kelvin in Buccal and EC50 in ELISA, we also performed the definitive Buccal affinity measurement for the clone 90G12F8 using multiple concentrations of Keytruda. As you can see from the data, the antibody showed a KD of 2 picomolar, which further demonstrated the high affinity of antibodies generated from the MonoRab platform. We compared the antibodies generated from stabilized hybridoma cells and recombinant expression. We found that to see whether they show any binding difference. As you can see from the data, for the 3 representative rabbit anti-Keytruda and idiotype antibodies, they all showed consistent binding profile between hybridoma production and recombinant production. In the last few slides, I will give you a quick overview about GenScript and our facilities. So GenScript is a world-leading biology CRO founded in 2002 in New Jersey, and currently is the #1 in the world for gene synthesis with over 1,700 employees worldwide. GenScript is publicly traded in Hong Kong stock market in 2015 and is specialized in antibody drug discovery and development. So in this slide, I will give you an overview of our facility. GenScript has over 13 years experience in antibody production. Our animal facility is both ALAC and ORO certified, hosting more than 8,000 rabbits and 12,000 rodents, and providing a broad range of antibody service from individual cases to bulk orders. We are equipped with high production capacity and have successfully delivered over 100,000 monoclonal projects, 14,000 monoclonal projects, and over 200 therapeutic lead generation and over 60 lead optimization projects. So if you have any antibody production need, GenScript is the one-stop shopping place for you. This is an overview of the antibody service we offer at GenScript. So besides the antibody drug discovery related service, which include therapeutic antibody discovery platform, anti-idiotype antibody generation, surrogate antibody, and immunogenicity assays, we also offer reagent antibody service for polyclonal, monoclonal, phosphor-specific antibodies, neutralization and blocking antibodies, as well as antibody pairing. We offer more than 100 catalog antibodies, diagnostic antibodies, and immunoassay development, such as ELISA kits for endogenous protein detection, host cell protein detection, PK, and immunogenicity evaluation. With the mission of make research easy, GenScript values clients' confidentiality and intellectual property as highest priority. We have a very well-established IP production system from of confidential agreements, secure online information transfer, IP and confidentiality trainings, compartmentations of clients' information to closed system with firewalls, password, and email control, and data backup. Notably, our IP production system has been audited and approved by numerous world-known biotech and biopharmaceutical companies. So I have discussed general rapid mAbs and also GenScript MonoRab Platform. So to quick summarize, choosing GenScript MonoRab Platform to generate your high affinity and high specificity rapid mAbs. In the MonoRab service package itself, we deliver antibody sequence to avoid stability issues of hybridoma and deliver recombinant antibodies to reduce reproducibility concerns in this timeline with most competitive price. So together with GenScript's overall strength as an integrated CRO, we provide one-stop service for customers, including peptide antigen synthesis, protein antigen expression, recombinant antibody production, even stable cell line production, and so on. So GenScript has mature and efficient hybridoma platform with more than 13 years experience of reagent antibody service and therapeutic antibody discovery. So as an important application for rapid mAbs, an endotype antibody service has achieved 100% success rate in GenScript for over 200 projects for client PK and immunogenicity studies. And definitely we can use this MonoRab to generate an endotype antibodies to support our clients or for all those PK and immunogenicity studies and for other challenging targets such as small molecules, such as for IHC applications, and also for other difficult targets. So with that, I would like to thank you for your attention, and I hope you have learned something useful for your antibody projects. So I will pass the ball to Elizabeth for the Q&A session, and I'm happy to answer your questions. Thank you.
Thank you very much, Dr. Yin. Yes, feel free to chat your questions for Dr. Yin in using the chat panel, which you'll find on the lower right side of your screen. You just need to type the question into the white box right next to the send button. Just above that, choose all panelists and click on send. Let's get started with our questions. In general, for which applications do rabbit monoclonal antibodies provide better solutions?
Yeah, that's a very good question. Actually, as I already mentioned, so for those, there are two kind of applications. One kind is for the regular applications, such as ELISA, IP effects. Usually the rapid mAbs, due to the high affinity, high specificity, and high diversity, can work better. So it can be as an alternative option for marine antibodies. But for some particular applications, such as generating antibodies against small molecules, against some very small different antigens, such as against different isoforms, or against anti-endotype antibodies for PK studies, and against for the IVD targets for IGC application, the rapid mAbs are perfect and are better option for those, specifically for those applications. But in general, rapid mAbs in the reagent detection field offers a big advantage over the marine antibodies.
All right, thank you so much. Our next question, what are the major concerns of using rabbit mAbs for therapeutic antibody discovery?
Yeah, this is a very interesting question. So as I just mentioned, so rapid monoclonal antibody is perfect for reagent, for detection, for all those purposes. But for therapeutic, the major concerns. So the one of the concern, as I mentioned, for the structure. So the structure of rabbit antibody is very different from that of marine or human antibodies, which will cause some immunogenicity and developability issues for the rabbit mAbs for therapeutic application. So this is the major concern, that the rabbit mAbs has some kind of developability or immunogenicity concerns for therapeutic indication due to the difference between the rabbit mAbs and the marine antibodies and the human antibodies. So they have difference, but actually those differences contribute to their high affinity and high diversity. So that's why they are perfect for reagent detection. But for therapeutic, it's kind of limited. But I have to say that to date, even though there is no therapeutic antibody drug approved originated from rabbit mAbs, but there are several in clinical trials, in clinical phase 1 and phase 2 trials. So they do have some advantage, and just there are some limitations.
And a question from Wei, may we know the name of your fusion partner cells?
We use a mouse fusion partners. We use a mouse fusion partners and optimize the fusion partner. And we use it as our trade secret, so I cannot tell you specifically which mouse fusion partner we use. But we use a mouse fusion partner, and we optimized the fusion conditions and optimized all the parameters during fusion. And we also use electrofusion instead of the classic chemical fusion. We use electrofusion that we optimized, also optimized for our marine hybridoma generation.
All right, perfect. Then we have a question from Samantha about the basic science behind why this system works. She asks, do you know why the rabbit immune system produces such high affinity mAbs and elicits an immune response to difficult antigens that a mouse would not respond to?
Yeah, interesting. So actually, in general, the robustness of the immune system is proportional to the size of the animal. So the rabbits, because they have a bigger size, then they have a more robust immune system compared with the mice. So that's a simple explanation. To go to more mechanisms, because in the rabbit monoclonal antibodies, there are more somatic mutations happened, and due to the nature of the light chain, especially in the light chain CDR3, there are so much diversity there, and its polymorphism is very large. So this all contribute to the robustness of the rabbit immune system, and also contribute to the high affinity and the high diversity of rabbit mAbs compared with marine monoclonal antibodies.
Yes, that makes perfect sense. Our next question, what is the difference between GenScript's MonoRab platform and the AbCam RabMab platform?
Yeah, that's a tough question. Actually, it's pretty straightforward. So AbCam, they use a rabbit fusion partner and it was patented, and we use a mouse fusion partner. But the key difference that we use our technical rules, we use the sequencing and recombinant antibody instead of from the stabilizing the hybridoma. So that's the key difference.
We have a question. If you inject rabbit monoclonal antibodies into mice for in vivo studies, what is the half-life or immunoresponse associated with that?
Yeah, so for the half-life, because there are several rabbit antibodies, they are already in the clinical trials. So the half-lives, they are close, and they are even better than the classic chimeric or humanized or fully human IgGs. But they do generate the immune response associated with that because, as I mentioned, they are not that similar with mouse and human IgGs. But you can still perform the humanization optimization for rabbit mAbs. So you can still perform the humanization. But still there is concern. Even you perform the humanization, there still is concern for the immunogenicity. But it's pretty stable. So rabbit mAbs are very stable because they have those disulfide bonds and they have those very well matured IgG structure.
Okay. All right. What contributes to the large diversity of rabbit mAbs?
Yeah, so for the largest diversity, there are the major diversity that comes from the light chain. As mentioned, there are two types of rabbit light chain, both the kappa and the lambda. And the kappa, they have both K1 and K2. And we have observed huge diversity in the K1 and also the K2 light chains. So the polymorphism in those K1 and K2, as well as the lambda light chains, especially in those CDR3, they contribute to the majority of the diversity of the rabbit monoclonal antibodies. So to answer this question, in short, the major diversity comes from the huge polymorphism in the light chains.
Thank you. And our final question, what are the major challenges to generating anti-idiotype antibodies?
There are several challenges for generating anti-idiotype antibodies since we have been working on anti-idiotype antibodies for a long time using mouse, and they are also pretty good. But rabbit provide a better option. So the major challenges, there are three. The first one is the CDR regions of antibodies are not that antigenic. And the second one is the percentage of specific anti-idiotype antibodies in serum is very low because most of the antibodies generated against the FC region. And the third one is we must optimize the clone selection for specific anti-idiotype antibodies such as to screen out those specific, to screen out those non-specific, and also to screen out high affinity and idiotype antibodies for good PK studies. So these three parameters contribute to the challenges. So to summarize, the low antigenicity of CDR region, the low percentage of anti-idiotypes in serum titer, and the clone selection for specific anti-idiotype antibodies.
All right. Thank you so much. Well, we've reached the last of our questions, so I would like to very much thank Dr. Yusong Yin for his presentation today. I'd like to thank everyone at GenScript USA for sponsoring today's web symposium. And most of all, I'd like to thank those of you who came and spent some time with us. We're very grateful you attended, and we hope you gained some information that will help make your job a bit easier. So with that, thank you again so much. Have a great day.
Thank you. Have a nice day. Bye-bye.